So let's look at another interesting study to determine how frequently heritable cancer mutations can be observed amongst cancer patients. And in this case, Illumina cancer panel was used. This is a very well established panel amongst 581 patients. And what's interesting about the study is that all of these patients actually fulfilled guidelines criteria for BRCA testing. So what was found? Approximately 18% of this population showed heritable cancer predisposition mutations or germline mutations in their genomes, and almost 13% of that were actually mutations in the BRCA1 and 2 genes, and between 5 to 6% of the mutations were observed in other genes. As just like in the other studies I demonstrated a moment ago, approximately 7% of the mutations were large structural variations involving large segments of the DNA. And what's interesting about the study is that they also focused on splice site mutations. And almost 9% of all the patients showed splice site mutations. So what are those? Please recall that I mentioned earlier that genes themselves are composed of fragments called exons. They're like pieces of Lego that can be shuffled in order to create different proteins or those different molecular products. And splice site mutations are actually mutations in between those gene fragments, as highlighted here with the red arrow. So let me show you some of examples from that particular study. So with BRCA2 gene, what is normal is to skip those introns shown here between gene fragment 18, 19, and 20. So 18, 19, and 20 fragments are supposed to be placed together, but because of the mutations, fragment number 19 was completely missed, and that resulted in a protein that wasn't working properly. So an equivalent of this would be if this type of protein or little molecular robot is normal, shown here, then imagine if a splice site mutation occurred and suddenly a whole chunk of the protein is missing. So you can imagine this could lead to disrupted function. A couple other examples found in other genes are shown here in PALB-B2 gene and CHEK2 genes, and both of these genes were also previously shown to be involved in hereditary breast and ovarian cancer predispositions. And that's important because cancer genes show splice site mutations about 1.5 times more frequently than what is typically observed on average within any other genes. Or another way to put it is that amongst most frequently mutated genes for splice site mutations, almost 1/4 of those genes are involved in cancer development. So let's look at a real-life example of cancer patient sequencing in Memorial Sloan Kettering Cancer Center in New York. And in a period of about 1.5 years, they sequenced more than 10,000 cancer patients for their tumor DNA. And amongst those 10,000 patients, approximately 10% of them were also sent by their doctors to be sequenced for germline mutations. So that means sequence for cancer predisposing mutations in their inherited genome. So what was found amongst those patients? Just like in the previous study I demonstrated a moment ago, approximately 18% of these patients showed germline mutations. And what's interesting is that, and this is important is to note, is that about 15% of those patients showed mutations in a single gene, but about 3% showed mutations in more than one gene. So that means there were more than one gene affected that resulted in heritable cancer predisposition. That's important because for a doctor to know that, it can dramatically influence potential medical management. Furthermore, this germline mutations we refer to as actionable. And what that means is that there's actually some form of prevention or therapeutic targets available immediately for these type of patients. In addition, amongst these patients with actionable mutations, more than half of them would actually have not been eligible for testing because they would have actually failed the guidelines criteria for testing. And almost 3/4 of all these patients showed mutations in DNA repair genes. Those are the type of genes such as BRCA1 and BRCA2 that we've been talking about so far, involved in single-stranded or double-stranded DNA breaks. So approximately 1/3 of them was BRCA1 and 2 mutations. Furthermore, about 20% of these patients who demonstrated germline actionable mutations had some form of therapy available to them right away. So that means based on the mutations that were uncovered, these patients had medical therapy management available to them immediately. Most common example of that were PARP inhibitors that I will be talking about shortly. Finally, of the patients who actually did have targeted therapy available to them because of DNA sequencing, once again, almost half of them would not have been able to actually meet the criteria for testing. That means about half of these patients would never had this medical treatment option available to them if they actually followed established guidelines for whether they are eligible for testing or not. So this shows you the benefit of DNA sequencing for any cancer patients. Furthermore, for almost half of all the patients who were eligible for targeted therapy, they had other family members tested who also showed similar mutations in other family members. So that shows you that when you sequence a cancer patient and discover heritable mutations, this can lead to cascade testing for other family members as well. And that means these family members can enjoy similar treatment benefits and potentially life-saving treatments that otherwise they might not have ever been aware of these type of mutations in themselves. So let's look at another study, this one involving 440 patients. But what's unique about this study is that all of these patients demonstrated multiple primary tumors, meaning they had multiple cancers throughout their life. And also they've already all been tested previously for cancer predisposition genes. And they all showed negative results. So this study was undertaken by the Cambridge University in UK. And as I previously already alluded, typically about 18% of cancer patients can be expected to show results indicating heritable cancer predispositions. So these patients, because they previously showed no such results, they were queried with whole genome sequencing. So this allowed to provide an opportunity to see if whole genome sequencing can demonstrate any further benefits than what would be expected from gene panel in cancer analysis. So amongst these patients, whole genome sequencing uncovered additional 15% of germline heritable mutations that previously were not discovered at all by gene panels. So that's actually quite a significant number. About 3% of those were double mutations. So once again, we're talking about mutations in more than one gene. You can imagine how complex it might be for a doctor to deal with cancer treatment if the actual cause of cancer is due to mutations in more than one gene. And only about 1.5% of these patients showed structural mutations, meaning those large segment of DNA mutations. This is in contrast to what I have previously demonstrated with about 7%, but that perhaps is not surprising. And the reason why is because these patients have already been tested with gene panels, and all the common structural mutations would have been potentially uncovered previously. So these cohort of patients was particularly devoid of structural mutations. But the take-home message here is that whole genome sequencing uncovered additional 15% of mutations that gene panels failed to recognize previously. So in total, we're looking at about 33% of patients could provide results if they were to be sequenced using whole genome sequencing, meaning the entire DNA is being analyzed as opposed to only selected genes, as what is typically done with gene panels. And because these patients also showed more than one type of cancer throughout their lives, this allowed to also ask question, what are the most common tumor combination types? And this is demonstrated in this diagram where different cancers are color-coded, and the breast cancer is shown in a lower right corner. You can see that breast cancer was the most frequent cancer observed amongst these patients, and you can see what were the most common secondary cancers for breast cancer patients. So number one amongst breast cancer patients, the second type of cancer developed amongst these patients was colorectal cancer, with approximately 6% of patients, followed by 4% of patients developing skin cancer and 4% developing ovary cancer. Likewise, for ovary cancer patients, the vast majority of ovary cancer patients develop breast cancer as a secondary cancer, followed by endometrial cancers.